that would be an alternative splicing where we skipped exon 2. You might also have an alternative splicing where you go E1 to E2 to E4, or E1 to E3 to E4, or E1, I think I already did that. Anyway, you get the idea. Now, one thing about this kind of alternative splicing is that you never change the order of the exons. You can skip exons, but you never change the order. So we might even splice from E1 all the way down to E4. And what's happening is at the junction at the start of every exon, this is referred to as a splice donor. And at the other end is the splice acceptor. So the idea is that you can splice from any splice donor to any splice acceptor. And so we could go from this splice donor to that one, that would be the most typical, or we could go from this splice donor to that splice acceptor, or from that splice donor all the way down to that splice acceptor, right? In general, with this kind of alternative splicing with skipping exons, you start with the first exon because the promoter's here, and then the changes start happening with these splice donors and acceptors, so that the first exon is almost always included. And what you're skipping are some of the other exons. So that would be skipping exons, and that's one kind of alternative splicing that occurs. Now what I want you to think about here is that these alternative splicings are going to give you different proteins, right? They're going to give you related proteins, but they're going to be different. The average human gene is spliced, I think I'm remembering this number right, is spliced in the average of about 4 to 5 different ways. That would be the average. Some genes have no alternative splicing, some genes have a lot of alternative splicing. And so, what this means is that even though we have a paltry number of 22,000 coding genes, coding for proteins, that the number of possible proteins that can be produced from those 22,000 genes may be as high as 100,000 or 200,000 because of alternative splicing. So this is helps us to partially understand how really complex eukaryotes can get by with not that many more genes than really simple eukaryotes. So, for example, Drosophila has, I think I'm remembering right, around 16,000, 17,000 genes for the fruit fly. We don't have that many more genes than a fruit fly, but we do more alternative splicing, so we produce more different kinds of proteins. So, alternative splicing helps us to understand partly why complexity of organisms doesn't result in sort of a concomitant increase in the number of genes, right? And that's because if you can do a lot of alternative splicing, you can make a lot of proteins, a lot of different proteins without making more genes, right? I mean, you could imagine that we could make these two different products by simply having two different genes, right? One gene that has exons 1 through 4 and one gene that has exons 1, 3, and 4. You could have two genes for doing that, but we don't need that. We can do it with one gene and just splice that one gene differently. So alternative splicing gives us a lot of possibility in terms of producing variable but related proteins. Now, this isn't the explanation, it's not a full explanation for why introns and exons are so popular in eukaryotes, because we have a lot of genes that have multiple introns and exons that don't get spliced alternatively. So not all genes do this, a lot do, but there are a lot of genes, again, that have introns and exons that don't get alternatively spliced. So it's not an explanation for all of the intron-exon variations we see out there in nature. Yeah.
Is the alternative splicing regulated, or is it?
Yes, it's regulated. So what you see for a lot of alternative splicing is that for a particular gene, this might be the way it's spliced in liver, and this might be the way it's spliced in spleen. And the liver and spleen are producing similar proteins, but variants of one another, and this one is optimized for whatever it's supposed to be doing in the liver, and this one's optimized for whatever it's supposed to be doing in the spleen. You see this with a lot of muscle proteins. So proteins in muscle get spliced in a lot of different ways depending on the muscle type. And some of the diversity is really remarkable, where some genes are getting spliced 50 different ways. You see the same thing in the nervous system, genes expressed in the nervous system, where genes are being spliced in a crazy number of different ways. So some genes have 50 or 60 alternative splicings known, and some genes have none. So interesting. Doesn't completely explain why we have introns and exons, although it certainly helps us to understand that. Other questions? Yeah.
So just different pathways bind to the spike neurosome and influence how it takes the effects on.
Right. So the way that splicing occurs, just to give us a little more detail, is that there's this big structure called the spliceosome that I mentioned before. So the spliceosome, again, is a big molecular machine like the ribosome that's made up of about half RNA and half protein. And the spliceosome is not nearly as well understood as the ribosome, but we're getting there. Really complicated molecule with 50 or 60 different proteins, a bunch of different RNAs. These proteins, some of them can come and go, and we know they're involved in regulating splicing. So splicing can be regulated based on what proteins are bound to the spliceosome. And so you could imagine that splicing occurs one way in the liver because there's a protein produced in liver that causes it to be spliced in one way, that's part of the spliceosome. Whereas in the spleen, that protein's not present, but there's another one that is bound to the spliceosome that causes it to splice in a different way. So splicing, we know, is regulated. It's different in different cell types, and it's regulated by proteins that bind to the spliceosome. And it's kind of analogous to what we do with transcription factors, right? How the spliceosome splices depends on what proteins are bound to the spliceosome. Now, the way the spliceosome works is that the spliceosome gets on to the 5' end of the pre-messenger RNA and works its way down doing the splicing. And how it splices exactly is going to be dependent on those additional protein factors. Okay, so we feel pretty good about that, because that's not the only way alternative splicing occurs. Another way is that you can have alternative splice donors and acceptors. So the standard splicing here would go from this splice donor to that splice acceptor, but exon 2 might have an additional alternative splice acceptor here. So an additional splice acceptor there. And in that case, the splicing could go from this splice acceptor, from this splice donor, to that splice acceptor. And that's going to make a different exon in the product, and again result in a different gene, a different protein product, sorry. So variable splice acceptors. An exon can have more than one splice acceptor. So The normal one is here, but there could be an alternative one there. And so again, we splice from here to this point as an alternative. And again, that's regulated. Okay? So we can have alternative splice acceptors. We can also have alternative splice donors. Instead of splicing from here to there might be an alternative splicing donor here, and you could splice from there to there, right? So an alternative splice donor. And again, what splice donor is used, whether it's this one or this one, would be regulated by proteins bound to the splices length. Right? So, when you look at the way genes are spliced, it's really quite remarkable. So the alternative splice acceptor and donor, it seems okay? Right? It gets even weirder. Genes can have more than one promoter. And so, I've shown that as the promoter for this gene, but some genes have an alternative promoter, and the alternative promoter might be located here. So we'll call this P2, we'll call this P1, and again, and this would be due to different transcription factors. Because of the transcription factors present in a particular cell type, one particular cell type might use promoter 2 instead of promoter 1. And if it uses promoter 2, it's only going to transcribe this part, and then that's going to result in a different protein also. So that's not really alternative splicing, that's really alternative promoters for the same gene. But it's still giving us an additional possible set of proteins from the same region of the genome, from the same gene region, right? So a gene in a eukaryote is a complicated thing, right? And it's pretty dynamic. In prokaryotes, bacteria it's not, right? The gene starts here and ends there. But in eukaryotes, it's not that simple, right? I mean, we can say the gene starts here and ends there, but certainly not all that region is expressed, and the way that gene is expressed is sort of all over the map with alternative splicing and alternative promoters. So you can't, in eukaryotes, you cannot think of a gene coding for a single mRNA coding for a single protein, because that's just not how it works, right? You've got a gene can code for multiple mRNAs, can code for multiple proteins, right? So an amazing amount of diversity. And if you look even at the human genome that is being, that's been studied more than any other eukaryotic genome, we're still, every year when I look at GAPDH or I look at TTR, the genes we work on, often I'll look at it for the first time in a year and see that there's a new, one or two new alternative splicings that have been discovered. Because remember, for a gene that's expressed in a lot of different tissues, in order to understand how that gene is spliced under all circumstances, you'd have to study how that gene is spliced in all those different tissues, right? And you also see different splicing over different stages of development. You also see different splicing over different environmental conditions. Wow. So are we ever going to understand all the alternative splicing going on? I don't know, because you'd have to look at the splicing that occurs in every cell type, at every stage in development, under every environmental condition, and only then would you be certain that you've seen everything, right? So I'm not sure that we'll ever know. But it's certainly complicated, and we know about the alternative splicing of a lot of genes. Now, a lot of genes haven't been studied in that detail yet. So when you look up genes and you look up what we know about their transcripts and how they're spliced, you got to take it with a little grain of salt, because it gets updated all the time, and it's going to keep going like that, right? So when I look at the way GAPDH is spliced now, compared to when we first started doing this experiment, it's more complicated now than it was then. And it'll probably keep getting more complicated. Okay, yeah.
Two things. Is there a specific term for the proteins that bind to the spliceosomes?
So it's not as standardized, but mostly they're referred to as splicing factors.
And then the other question, for the alternative splice donors and acceptors, will those also be surrounded by those GU and AC sequences?
Yes, so they tend to be. Now, at these junctions, at these splice donor and acceptor junctions, there's this GU-AG rule, but there are other nucleotides, both in the exon and the intron. So other nucleotides in this region that you tend to see more frequently than others, but it's not a set sequence. So the idea is that whatever the sequence is here at this splice donor, there's another sequence here that's very similar to that, and so the cell can use either one, again, depending on what splicing factors are present. Okay? Really complicated. So I always tell my students, if you want simple, study bacteria. If you want complicated, study eukaryotes.
Are we going to learn about some of these databases where we can
Yes,
look up?
yes. So we're gonna start working with our computers pretty soon, and we're gonna go to the databases, and I'll show you for GAPDH and TTR. I'll show you the alternative splicings that are known. Yeah, look at that.
I don't know if there's an answer to this, but it's a little strange that there's so much DNA that does nothing, that doesn't code for anything. But the DNA that does code for something is used for like many,
A lot of different
things. So it's very efficient, but then there's also a lot of stuff that's not being used at all. Are there any guesses as to why, what that optimized for?
Well, so I can, yes, we can make guesses. I would say that that the driving force for developing different proteins by alternative splicing as opposed to generating a new gene is that alternative splicing can probably evolve faster. It's easier to switch how a gene is being processed than it is to evolve a whole new gene, right? So I'd say from an evolutionary standpoint, this probably can evolve much faster than the development of new genes. So I think probably the evolutionary pressure in generating complexity in higher eukaryotes has pushed things in this direction. But again, we have a lot of genes that have multiple introns and exons that aren't being spliced alternatively, at least as far as we know. And so, yeah, I don't think we know everything yet. Clearly, we don't know everything yet.
Are there intron sequences of the pre-mRNA? Are they ever found to be like regulating mRNA structures?
Yes. So if you look at introns and exons, you might think kind of naively, you guys are smarter than this now, but you might think naively that only mutations in exons would have an impact on the protein in terms of its function. And that ends up not being true. You can have mutations in introns that impact how much that gene is expressed. In fact, you can have mutations in introns that block expression altogether. Well, how is that possible? Well, the thing you have to think about is that your RNA is a single-stranded molecule, and it's going to fold up in a certain way. Every RNA folds up in its own way. And the way it folds up is, we know, important for splicing. If it folds up the wrong way, it won't get spliced properly. And so because of that, a mutation in an intron can change that folding, screw up splicing, and then be detrimental to the function of the protein and the survival of the organism. So mutations in exons certainly are, in general, more problematic, but there are mutations known in introns that are also extremely problematic for expression of the gene. Mutations at intron, in donor and receptors are problematic, but some mutations right in the middle of an intron can cause a reduction in expression of the gene or completely stop expression. So we need to think about folding of the RNA as being an important part of splicing, which of course then is critical for making the protein. If you don't splice this thing correctly, or if you don't splice it at all, then it's not going to be functional. So, eukaryotes, complicated,
So
right?
do eukaryotes have these regions that are like terminators?
Oh yeah, so thank you for bringing that up. So remember in our nice bacteria had a nice promoter, and then it was all coding, and then there was a nice terminator, and we didn't have alternative splicing, and things are, pretty easy to understand. Those terminators were great because transcription starts at the promoter, ends at the terminator, our transcript is that big, right? Really straightforward and easy to understand. In eukaryotes, the reason I put the arrow here is because transcription starts at the promoter, goes down the gene, but there's no real terminator down here. So polymerase kind of dribbles on past the last exon, and it might terminate here, or it might terminate here, or it might terminate here. So there's no real set terminator. Sometimes it is. So eukaryotes, again, complicated. Sometimes there's a sequence that's like a terminator where the polymerase tends to fall off, and that's usually it. But then there are genes where there are things that sort of look like terminators where, 10% terminates here, 10% there, 10% there, 10% there. So there's no set in the transcript. Now, in the process of RNA processing, what happens is that there is site. After the last exon, there's a site where an enzyme cuts the RNA, and that's where the poly A tail is put on. So even though the pre-mRNA has no set ending spot, after this cut's made and the poly A tail's put on, then they all have the same 3' end. So the transcripts after processing, they're going to all end at the same spot. And that's because there's this site where you have the cut and then the poly A tail is put on.
The number of A's in the poly A, is it variable or?
Yeah, the number of A's in the poly A tail, kind of the set thing you see in the literature is 20 to 200 A's. I'd say that's pretty soft because as soon as those A's get put on, you can start getting A's lopped off. So one of the arguments about the 5' cap and the poly A tail is that they protect the transcript. I think I mentioned this before. The 5' cap looks weird, and ribonucleases don't know quite what to do with that, so that helps protect the 5' end. The fact that you've got this long poly A tail at the 3' end also helps to protect the transcript because you have nucleases that take off nucleotides one at a time, and so if you've got this long poly A tail, it's going to take time for that poly A tail to be removed before you start getting where the action is, right? And so the idea is that both of those features help protect the RNA, and that partly explains the long half-life of mRNAs in eukaryotes compared to bacteria.
Is the poly A site where it gets cut, like a general site across all mRNA, like a specific sequence that was for?
Yeah, it's specific sequence, AU, AAA, or something like that. It's called the poly A addition site. Poly A addition site. And there's an enzyme that cuts there, and there's a specific polymerase that adds the poly A tail. It's called poly A polymerase. It's an RNA polymerase, but it only adds A's. else? So it's complicated, there's nothing simple about it. And if you wanna study a particular gene in eukaryotes that's spliced alternatively in different tissues, you could have a whole career doing that one gene, and people do it. I'll give you some examples later of sort of extraordinary human genes that are really big and spliced in different ways. I'll give you the numbers later, but it's pretty entertaining. Okay, so that's what I wanted to tell you about alternative splicing for now. We'll talk about it a bit more when we do some computer work later. But I want to go to the protocols now because I want to make sure we have enough time to do everything.